Hi guys, welcome to the channel. I am Matangi and today we are gonna see about an online ligand binding site prediction server. So first let me explain about what is a ligand binding site. So here I have taken a picture from the internet. Okay, in the picture you can see in the left side you can see the protein molecule which is in the cartoon representation with its ligand binding site residues in the red color, and in the right side you can see the same protein with the same amino acid residues, the ligand uh, binding site residues interacting with the ligand molecule which is in green, I'm sorry, which is in a blue color, and in this picture's case the ligand molecule is a glutamine. For instance, take a any protein or any molecule as their target molecule, and the molecule, the one which comes and binds to a target molecule, is your ligand molecule, and your ligand can be possibly any protein molecule or any small molecule, preferably any drug compound or something. So when such ligand compound or ligand molecule, it comes and binds to a target protein or target molecule, it binds at the specific site of your target protein and interacts with the amino acid residues at that specific site, forming hydrogen bond interactions, hydrophobic interactions, salt bridge interactions, and so on. So when such ligand molecule, it comes and binds to your target protein at the specific site and interacting with the amino acid residues at that specific site, that site is your target protein's ligand binding site. And why should a ligand come and go and bind to your target protein? So in the picture, in the left side and the right side, you can see the same protein structure, but there's a difference in their structural conformation. You can see the difference before the protein I mean without the ligand present and with the ligand present. So when a ligand molecule, it comes and binds to a target proteins in the ligand binding site, it just tends to change the structural conformation of your target molecule so as to either activate it or inhibiting it. So the ligand molecule, it comes and binds to a target protein either to activate your protein or to inhibit your protein by inducing the structural conformational changes. So that's why ligand is binding to a target protein, and the activation and the either activation or the inhibition part is totally dependent on to which protein and to which ligand. So it is totally dependent on the protein and the ligand molecules. So now we will see about the web server. So today I'm gonna talk about the web server, PrankWeb server. So why I have chosen this server is because this server is one of the servers that got cited in many research articles. That means citation is like many research works, in many research articles they have used this web server for their research publication and for their research work. So that's what citation is mean. And in the recent coronavirus period, this is the article that is related to the coronavirus SARS-CoV-2. So this, in this article, they have also cited this PrankWeb server, and this article is published in the September 2020. Vitamin D is a potential inhibitor of COVID-19 in silico molecular docking to the binding site of SARS-CoV-2 endoribonuclease NSP15. So this is the article. So I will give both this article and this article's links in the description in the description box, and you can see this prank web server. This research article is an exclusive research article of this prank web server, and in this article they have mentioned how this server, it predicting its ligand binding sites, how it's the way of how it's predicting its ligand binding site of the proteins, and how to submit the form, and how to do the results analysis after the results have were published. So they have mentioned so many things about this server in this research article, so kindly go through it. So let's take a look at the PrankWeb server and job submission. So just go to PrankWeb server, just type. So here you can see it's the PrankWeb server. So now we can see the job submission form for the ligand binding site prediction. So you can either mention the PDB code, that is if you want to find a ligand binding sites of any already available known structure which is in the PDB, so you can mention that PDB 4-letter code, or else if you are having your own files like modeled files, protein files or something, just choose file and upload your files, and just mention if there are any changes that you want to um, for which you specifically want to find the active sites. I mean, sorry, active sites not, it's not, it's a ligand binding site. So if you want to find to any specific chain the ligand binding site, just mention the chain, and at the end you can see the conservation analysis. Yeah. So if you want to make any conservation analysis with already known structure which is available in the PDB, so just mention your 4-letter PDB code, or if you have any multiple sequence alignment files, just upload it, and these both options are optional, so you can either upload it or you leave it, so it's totally up to you. So once you have done all the thing, you just submit, click the submit tab here, and after submission, and in this form, in this form filling, there's nothing they have asked about any email address or something, so there would be no notifying of results from this server to our email id. So after you click, after you clicking the submit tab here at the end, you can see here in the URL side they have they will give the results link, so just bookmark the link, or either what, just copy and paste the link in the notepad or something and just save it, and often come and visit this server whether the results were published or not, to know whether the results are published or not. So just save the results links. So once after the results were published, your result page will look like this, and here I have already submitted the protein structure, and I already submitted the protein structure which I took from the PDB. Yeah, it is already known protein structure, and it is the 1M7Q, that is a MAP kinase, and the PDB code is 1M7Q. Just go and take a look. So this protein, in this protein you can see the colors are indicating, the different colors are indicating the different ligand binding site predictions. So why I chose to submit the already known structure from the PDB, because I want to test this server whether this server it's predicting the ligand binding sites effectively or not. So that's why I chose to submit the already known structure from the PDB. And as you know, the 1M7Q, the protein in the 1M7Q, it is the complex structure, that is already known available complex structure, that is already with the ligand molecule in its ligand binding site. So I just want to make sure whether this server it's predicting a ligand binding site with the good accuracy or not. So that's why I wanted to test this server. So as I already told, this server is a, I mean this protein is already available protein. So I just opened this available protein in the PyMall, which I downloaded it from the PDB protein data bank. So you can see, as I already told, this is a complex structure, and this is the ligand that is already present in this 1M7Q complex structure in this 1M7Q PDB code. So you can see the ligand binding sites in the blue color, that is surrounding the ligand, and I have shown the ligand in the red color. So this is the ligand molecule, this is the whole protein molecule, and I have mention the binding site residues in the this ligand's binding site residues in the blue color. So now we can compare both the pockets. So here they have given approximately, I think, like 5 pockets and the pockets were scored ranked according to the pocket score, and also they have individually given the amino acids that are making up the individual pockets and the conservation score for individual pockets. So from this both already known structures, you can see that the blue color in the same in this server, the blue color is in the same site as in here in the PDB code, in the PDB structure. So this server, it can or it might or no, it is actually predicting the ligand binding sites with the better accuracy. So you can see from this proteins predictions. So if you want to know the binding site residues, just move your cursor and place the cursor of on any place on the protein. In the same screen you can see uh, they have mentioned the amino acid, the chain, and the amino acid number. For example, I'm keeping the cursor here, it's in showing the cursor pointed site in the yellow color. So on the above, you can see it is the glycine residue in the A chain and the glycine 110, and below that you can see the isoleucine A84, leucine A167, aspartic A168 and something. So you will get to know on how which residues are forming which individual binding sites by just moving the cursor and noting down. And also they have colored the other parts of the other sites of the protein. In some sites they have colored it in black, and then it goes from the dark gray to light gray, and further on to white color and all. So these colors are indicating the conservation regions, conservation amino acids in the protein structure. If it is so black, that is the amino acid is more conserved, and if it is in the white, it is there is no conservation of the amino acids, and this whole the black to dark gray, then to white gray, and light gray, and to white. So these are indicating the different levels of degrees of conservation of the amino acids in the protein structure. So if you want to download the report in the tools, on the right side you can see the tools tab. So under the tools tab you can see the download option. So just click download results. So in the at the end of the screen the results will be downloaded in the zip format. So once you have downloaded the zip format and extracted the results, you can see there are so many files. The most important file is this Excel file, and if you open the file, the Excel sheet will appear like this, will look like this. So in this Excel sheet they have mentioned the pocket numbers, rank, pocket scores, and the XYZ coordinates, that is the XYZ coordinates of the individual pockets, and the amino acids that are making up the individual binding sites, ligand binding sites. So these two, the coordinates and the amino acids that are making up the individual binding sites, these both things are important to we further proceed for the virtual screening process. And what is a virtual screening process? Is that we are screening or filtering the small molecules that are likely to bind and fit into your individual ligand binding site. So now after we once predicted the ligand binding sites, then we are going to do the virtual screening to find which small molecules are likely to bind to your individual ligand binding sites. So that's why we are doing the virtual screening. So this is how we are analyzing the results in which we got from the PrankWeb server, and it's also one of the good server, and it's cited in many research articles. So if you want to publish any or go for any research publication thing, you can surely go for this server, and it's one of the best server as I know. And yeah, that's it for the today's video, guys. Thank you so much.